Hello and welcome back to Six City Marketing. I'm Hannah, and today we have Dr. Mitch Drumm, a distinguished university professor at Case Western Reserve University. He's a professor in the Department of Genetics and Genome Sciences in their School of Medicine, and he's the director of the Research Institute for Children's Health. And that's what brings us here today. It's the Institute's 2026 Giving Kids Hope Gala, where they're raising funds for some incredible work toward treating childhood genetic diseases. So we'll hear from Mitch about all of this, how far genetic research has come during his career, some of the ways artificial intelligence is being used to help researchers today, and what motivates him to continue to make medical advancements and help families in need. So get comfy. We've got a heartwarming 30-minute conversation for you, and let's jump right in. Well, Mitch, thank you so much for being willing to have a conversation with me today. I'm very excited to hear about Giving Kids Hope 2026, and we're very excited to be involved as well.
I'm thrilled that John and his company of Six City Marketing are supporting us.
Absolutely. It's a great cause. How did you get started in genetics and disease research?
How much time do we have?
We've got all the time we need.
Well, I'll give you the long story, and this goes back to grade school, so take yourself back 60 years. My sister and I were riding our bikes and came across a guy in our small hometown who had mice that he was raising, and we each bought one from him. And as young as we were, we didn't know the difference between a boy and a girl mouse, and turns out we had one of each. And so in a few weeks later, we had more mice, and those became my fifth grade science project to track the coat colors of the mice. So that was my introduction into genetics. So this is back in the 1960s. Anyway, fast forward, and I chose genetics as a major in college, and then went on to human genetics for graduate school. And while in graduate school at Michigan, neighbors from that same hometown had a son born with a disease called cystic fibrosis. And so I started working on cystic fibrosis as a project, and about 4 years later, we actually found the gene that causes the disease, and I've been working on it ever since. So genetics has been a long, half a century interest of mine.
How did the Research Institute for Children's Health come about?
The Research Institute came about because we had made so much progress on one disease, cystic fibrosis, and much of the technology we use for that one disease could be applied to the thousands of other genetic diseases that are out there. And I sort of liken it to, if you have a bus that you got because you want to get to whatever place, you've got plenty of capacity for taking a lot of other people along. It doesn't cost another penny to do that. So we thought the same idea with the technologies that we've got here to work on cystic fibrosis, we could apply to many, many other genetic disorders. And so that's what sort of spawned the idea of the Research Institute, was to take all the faculty here working on different genetic diseases, bring us together, create an infrastructure so we can share ideas, share equipment, and then to reach out to the public and tell them what we're doing and hope that they'll support those efforts. The math behind what we're doing is the estimates are that somewhere between 1 in 10 and 1 in 30 kids will have some sort of a genetic disorder, right? Which sounds ridiculously high, but that includes things that are fairly benign as well. So color blindness might be an example where it doesn't really affect your health, but it's considered a disorder. Okay, but then you have other ones that where a baby may pass due to the disease shortly after they're born, and so it's a very wide range of things that make up genetic disorders. With that kind of an incidence, it means that every family has somebody affected, whether it's niece, nephew, brother, sister. Going back in my own history, if you look at your class picture in grade school, I think you can pick out those kids too oftentimes. It affects everybody.
You've got Giving Kids Hope 2026 coming up here, and it looks like it's going to be an amazing event. So what are you most looking forward to as August 29th approaches?
So our annual gala outreach and fundraising event we call Giving Kids Hope. And this year, this will be our 11th year doing this. And each year, I love the positivity of the event. We get a lot of people who get excited. Our lab research people get excited to see folks who are actually interested in what they're doing. So it's a very positive, reinforcing event from that. We get to talk to the community members, and they seem to embrace what we tell them, and we have an awful lot of fun at the same time. So it's a great example of having a seriousness of purpose, but it doesn't mean you can't have a good time as well. So it brings everybody together.
If somebody's interested in being a part of it, it may be too late to join this year, I don't know, but if they want to join at some point, where can they go?
If one were interested in what we're doing, they can always email me. It's mitchell.drum@case.edu. We have a Facebook page. We have a page through the university. So if you search under Research Institute for Children's Health, it'll come up with contact information as well. So we're pretty much sold out for this year's event, but we do a lot of different things. We provide tours if people want to actually come and see with their own eyes what we do with the funds we've raised. We'll walk you around, show you the facilities and how we do our experiments. We have other events. We have a supporter who's a yoga instructor, and so she carries out yoga events for people. We have another one we call Gene Targeting, that is a skeet shooting event where we get teams together and compete for who can hit the most clay pigeons, and then our gala, and always looking for other clever ideas of how to engage with different people.
Well, we'll include those links with the video, and it sounds like there's no shortage of ways for people to get involved if they want to help or they want to learn more.
Exactly. Yes.
Great. Now, I noticed that you're going to be doing a collaboration with UCC in Ireland. And what's that going to look like?
Yes. One of the things that I'm excited about this year is that we have been broadening our research arms, I guess we'll call it, to where each year we get more and more faculty at Case Western Reserve University involved in our program. But we also have developed collaborations outside of the university. And over the last couple years, we've developed a relationship with faculty in Cork, Ireland, so at University College Cork in particular. And two of those faculty members are actually going to join us at Giving Kids Hope. So they're coming to Cleveland, and we're delighted by that. And as an aside, Aer Lingus has a direct flight from Dublin, Ireland to Cleveland. It's the one international European flight you can get out of Cleveland. So we're happy that that's made it easier for us to make these connections.
Now, how many diseases is the Institute researching in 2026?
The Research Institute is working on, I'm going to say loosely, about 24 different genetic disorders. So we've started on a number of these. Some we're making great progress with, others we're reaching difficulties and things. But overall, we're, I don't want to call it dabbling, but we've started programs in about 24 different disorders.
Can you share a specific patient or family story tied to one of the 6 research areas?
I've had the humbling pleasure to meet a number of families with children affected by various diseases. And one of the first ones, in addition to our work in cystic fibrosis, where I probably met the most of these families, one of them have a daughter with a thing called a glycogen storage disease. And this is one where your liver doesn't make the enzymes it needs to metabolize glycogen. So normally you or I our liver would store up carbohydrates so that while at night when we need energy or we're exercising, the liver will break that down into glucose so your body has the energy it needs. This young lady was diagnosed when she was just a few months old, and I met her when she was 15. And she came in and was asking, a friend connected us, and I didn't know her background at the time. And this young, I think she was a sophomore in high school at the time, had the most pointed questions for me about biochemistry and genetics and so on, only to learn that she's affected by this disorder. And as I got to know her better, I got to learn that every 3 hours she would need to take a supplement, basically cornstarch dissolved in water, and she would have to take that every 3 hours, and that meant 24 hours a day she was on that 3-hour cycle. So her mom and dad had their phones set to wake one of them up every 3 hours so they could make sure she was getting what she needed, lest she essentially would be in danger of going into a coma if she didn't have that. And I bring that story up because anybody who has kids remembers that first year, maybe 2 years of having an infant where, yeah, you're getting up all the time. But now imagine your child's 15, 18 years old, and you're still having to be on that same schedule. And it's not just waking up because the baby's hungry. Now you're waking up because it's their life that's at stake. So, and I'm thrilled to say this same young lady is now at the end of medical school. So she's done quite well. So it's a great story, but she still has to do that same 3-hour regimen, even in her 20s now. So, and that's why, so we're hoping with what we're doing for genetic diseases will eliminate that need for her. It'll fix the gene that causes the disease and make her life a little easier.
Absolutely. Thank you for sharing that. Do you have any other stories or case studies that you'd like to share?
Another disorder we're working on with a local biotech company is one that's called a mucopolysaccharidosis, which is a bit of a mouthful, but it's one that where an enzyme is missing, and it causes a buildup of a biomolecule that chokes off the nervous system. And I got to meet the parents of a girl who had this disorder, and this daughter passed away from that. It was another one, a genetic disorder of these types. It is a 24/7 thing for the family to have to deal with. And mom and dad are still so passionate about it that they've been raising money to help us and others with the research to try to prevent other families from going through what they went through.
It's really inspiring to hear.
It truly is. I mean, these are folks that I cannot imagine what they've gone through.
Are there any roadblocks, or is there anything that you need to advance your work?
The biggest barriers or hurdles that we face for the research are hands to do the work and money to pay for it. And that's the reason for our fundraising events. The other thing is the traditional approach we use for our science, and rightly so, is we have to put together proposals that describe what we're planning to do. We send them to the government or to foundations, whatever funding agencies. They review them and we get feedback. And for most of the government ones, by the time that lapses from submission to when we actually get funding, if we get funding, can be a year and a half, 2 years from just the beginning of the idea. Right? Sometimes it's faster, but that's a fairly typical time frame. So I'm bringing that up because it doesn't take us a year and a half to come up with an idea. So we really want to get going as soon as we've got the idea. We want to do those preliminary experiments to see if it's going to go anywhere. And that's where the philanthropic involvement comes in, because now, because of the generosity of donors that have supported us, If we get an idea on Monday, by Tuesday, we're starting on it now. So it's really, really accelerated our ability to do things. And then we can get more information, more data that we can then go to the government and say, hey, I've got a really good idea and I've got evidence to show you that it's a really good idea. So it increases the likelihood that we'll get subsequent funds for it as well.
All right, now I'm very curious about the process of discovering that CFTR gene that causes cystic fibrosis, and I'd love to hear more about how that came about and who was involved.
My background in cystic fibrosis started as a graduate student, and when my neighbors had a son born, where I grew up, neighbors, when he was diagnosed with CF, I was working on a technique at the time. This is long before we had human genome information and things, and only a couple of disease genes out of the 20,000 genes across your chromosomes. We knew very little about any of them. And so we were really starting with what today would be considered rudimentary technology, so obsolete technologies. But what I was working on was, if you think of your chromosomes as these big, long, linear molecules, how do you get from point A to point B? And point B being a disease gene and point A being you don't know where. So it was a lot of unknowns, but we developed some techniques that would allow us to do that kind of jump along the chromosome and get to a different place. Then when I found out that these neighbors had a son with CF, I asked my PhD advisor if I could apply that technique to cystic fibrosis. And he said, sure, why not? We got to try it on something. And there was a group in Toronto that had just identified a DNA marker. I'm not sure how else to describe that, but a part of the genome that was tracking in families with cystic fibrosis that we knew had to be reasonably close to the disease gene. We just didn't know how close, and we didn't know if we needed to go left or right. So we took what they had and used that as a starting point and started moving both directions along the chromosome. And we saw that one direction got us closer to the disease gene, one got further, so we knew which way to go. Anyway, 4 years later, we actually found the gene that we then called CFTR for the cystic fibrosis transmembrane conductance regulator. Okay, another mouthful. And I bring up the time frame of that 4 years, because that is now something with today's technology that probably would take far less than a week to accomplish. So that's where we are now with things, right? And as another offshoot of that discovery, my thesis advisor is a guy named Francis Collins, and Dr. Collins went on to be the director of the National Institutes of Health and to lead the genome efforts there as well. And so what they were able to do starting in the early 90s was to map the entire human genome, so all 23 chromosomes, and look and see where pretty much all of the genes are across all those chromosomes. And that whole process took about 13 years and $3 billion to do. Okay. Now, with what has resulted from that technology is now to where we can do an individual's genome for far less than $1,000 in about 24 hours. So it means that we now have the ability to think about genetic information when we're putting together somebody's medical profile, right? That you could now go in and look and see whether or not, in theory, whether or not you're at risk for this disease or that disease and so on and so forth, right? That just like your blood type and your blood pressure, things like that are all part of your medical record, we could do that with your DNA as well. And I'm sorry, I'm getting on my soapbox a little bit here, but I think one of the problems with genetic disorders is that individually, they're very rare. So you'll see most of them are maybe 1 out of 50,000 people will have a particular one. And so that most doctors will not have seen a given disease in their lifetime. And so they often don't recognize it for what it is. It may look like something else. For a long time, cystic fibrosis was thought to be sort of a pneumonia or something like that that affected the lungs. Well, now we know it's a very specific disorder. So now imagine you could go in and just looking at somebody's DNA sequence, you could prevent a lot of misdiagnoses just by looking at their DNA sequence.
I'm amazed by all of this. I pulled a quote here from your bio that says that your work in the early 1990s showed that CF cells could be corrected by introduction of a normal copy of the CF gene. What was that like to make that discovery?
So that was a really a critical juncture for us, because at that point we didn't know what finding the gene was actually going to mean in terms of what it would do for patients. So you find the cause, but it doesn't mean you can fix it or do anything about it, right? So a tree falls across the road. Yes, it stopped the traffic, but that doesn't mean you can fix it. That's probably a poor analogy, but it was that kind of thing. You don't know how much damage is done and whether or not it's reparable. So when we were able to put a normal copy into a CF cell, a normal copy of that gene into the CF cell, and show that we could bring what was called the chloride channel function, the defect of the cell, that we could correct it, it said, hey, well, maybe we can do that in patients. Okay. And that turned out to be true, not the way we had envisioned it, but it turned out to be true that a number of years later, so that's the early 90s when that discovery was made, and then a company got involved and started using robotics and miniaturization to screen thousands and thousands of chemicals to see if any of those would restore that CFTR function, that the protein that causes CF. And lo and behold, they found one that does. And they were able to develop that and make it into a drug. And that drug now is being taken by about 80-90% of CF patients, and it fixes very much of that defect. And in some patients, they're getting better, and some it's preventing increased problems in the disease, and some it's just slowing it down. And there's still a small portion of the CF population that have mutations that don't make any of this CFTR, so there's nothing for the drugs to work on. So for them, we got to come up with yet a different strategy. The point being, it showed that you actually can make a clinical difference. You can change the health of these people by fixing that thing. And up until that point, CF and all the other genetic diseases have only been treated by treating the symptoms, because we didn't have a way to get in to the source of the disease. If they had an infection, we would treat it with antibiotics. That's not what causes the disease, but it's one of the symptoms that we could get at. To me, that's the biggie on this, was showed you actually can fix it. And I will mention also, there have been a couple of recent examples, one in sickle cell anemia, where now getting in and correcting the gene it's more complicated than that, but restoring the cell's ability to make a hemoglobin that will work is having an immense impact on some patients with sickle cell anemia. There was a story that many people have seen about it's called baby KJ in Philadelphia, who was treated for a liver disorder, where another one where an enzyme is missing, and they went in and fixed the gene mutation, and that child is doing remarkably well. What would have been a fatal disease is now one where he still needs treatment, but not nearly as severe as it would have been had they not been able to repair that gene. So these examples are starting to mount up, and it's just opening the door that once that first precedent is set, you get excited about, okay, now it's just a matter of time. So we've got 7, 8, 9, 10,000 different genetic diseases out there we've got to tackle, and we've got a handful of them that show that we can actually do it. So now let's try to put the foot on the gas and see how many more we can do. You asked about hurdles a little bit ago. One of them now is, okay, getting into the liver is probably the most straightforward. If I stick something in my blood, the first thing it's going to do is get passed through the liver. The liver cells are going to suck it up because that's what they do. They try to get rid of things that shouldn't be there or metabolize things. But what about if we need to get into the brain, the lungs, the intestines, muscle, whatever? We've got to come up with different strategies to get into those organ systems. And so that's where a lot of us are spending our efforts right now.
Okay, I'm curious, as you've been telling me about technological advancements that have happened and it's incredible how quickly things can happen now compared to things that would take years before happening, sometimes overnight. Have there been any advancements with AI of late that you're excited about?
Yeah, and AI is such a broad term, but it is definitely having impact in a lot of different ways. And I will tell you maybe the least interesting, but one that's most useful for me is using AI to help me find literature in a particular area. Before, you'd go through the table of contents of a journal and see if there's something of interest, and you have to do a lot of reading just to see if it's even a paper that's relevant. AI helps me go through that almost instantaneously and lead me to things that I can then evaluate. So I can go right to one of the sources. So that's one that many of us are using. But the other one is more of pattern recognition types of things that on the pathology side, let's take cancer as an example. If you're looking at a biopsy, if there's a tumor and it's big enough, a pathologist can pick that out. But if it's not, if it's still very small, or even as an MRI or a CT scan or one of the imaging techniques that they use to look for tumors, the computer now using AI has been trained to find those things at a pixel level far smaller than what our eyes can find. And the computer doesn't get tired after looking at hundreds of these images as well. So it can find things that we just can't. We have to train it to know what to see, but it can do that. And the same kind of mentality works now with our genetic data, where you've got 20,000 different genes that all have to make proteins that work together to make a cell do this and that and so on and so forth. And in my opinion, our brains are not capable of processing all the permutations those 20,000 things do to interact with each other, but the computer can find patterns that we just don't see. So the short answer is yes. I think as maligned as AI has been for certain reasons, I think it is massively helpful in the biomedical research arena. And I'm going to also say that it is not the end result. So it can pick out and predict things, but we still have to do the empiric experiments to show that it was accurate. So it's great for helping us speed up our idea development, but it doesn't prove things. We still have to do that with our pipettes and our scalpels and all the things like that.
That sounds like it's a useful tool, but it's not taken on more than it should.
No. And it doesn't, the computer does not do lab experiments. It just, it can pretend to, but it doesn't do them. Yeah.
Got it. I was wondering philosophically what your discoveries have told you about the universe and kind of the nature of reality, as you've learned more and more about genetics and just how everything works. Is there anything that you'd like to share along those lines?
That's a big question. One of the things that I guess comes back almost every day is every time we find something, it means we've got to dig in and learn more about what's already known. And so it's a constant educational process. And every day is a revelation of how much we don't know because it's, oh, I didn't know that. I didn't know that. And so for me, I love the learning part of it, but it's just so humbling to realize how much we don't know.
What are you hoping to accomplish in 2026?
I hope we can add to the number of disorders that we can develop therapies for. We've transitioned from the is it possible to an engineering phase where how we make it efficient enough to work. How do we get enough diseases to a therapeutic stage? And this is where we have to start working more with our clinical colleagues on setting up clinical trials for these types of disorders and seeing what difference we actually can make.
You started with some mice and you were basically breeding these mice and keeping track of their different coat colors, and that got you interested in genetics. And then you had a neighbor later on who had cystic fibrosis, and you were interested in that disease and understanding it and helping cure it. And what in all of this, what is it that motivates you?
I think it's curiosity. I always want to know how things work. And the other is I just grew up in an environment of wanting to help people. Mom was a social worker, dad was a teacher, and that's just been part of my life. And I've always had a passion for helping kids, whether summer programs, coaching, babysitting, whatever. And that's never diminished. So there's plenty of adult disorders too, but to me, the kids are they're our future. So we really need to push that.
Thank you so much for all that you do and for taking the time to meet with me today and share. And congratulations on selling out the event, Giving Kids Hope 2026. Love hearing about the success there and encourage everyone listening to go check out your website. We'll have all the links down below and get involved.
Thank you very much, Hannah.
My pleasure.